Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Francesca Telese from UC San Diego and Jessica Zhou from Cold Spring Harbor on the show. This time we are doing things a little bit different since we have two guests on the show. Please let me briefly introduce both of you to our audience. Francesca, you got your PhD from the University of Naples, Federico II in Italy in 2007. You then moved on to the University of California to do your postdoc. After your postdoc, you stayed there and to become assistant project scientist, and currently you are associate professor. Jessica, you did your PhD with Francesca from 2017 until 2023, and this is why both of you are in this episode together, and you are currently a computational postdoctoral fellow at the Simons Center for Quantitative Biology at Cold Spring Harbor Laboratories. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
Sure. This is Francesca Telese, and I became interested in biology probably when I was in middle school, and one of the professors at school showed us a book on the structure of DNA, and I borrowed that book for the summer reading, and that really turned my interest in this fascinating molecule that decides who we are. And that started, I would say, my career in biology. And then I did a PhD in neuroscience in Italy at the University of Naples, where I studied, I was more interested in Alzheimer's disease and how different protein complexes were implicated in the neurobiology of Alzheimer's disease. And that's where my graduate work led to an initial discovery of some transcriptional mechanisms that were implicated in Alzheimer's disease. And I wanted to learn more advanced methods to study transcription. And that's where I looked at different, pioneering labs that were working on gene expression regulation. And that's where I reached out to Michael Jeff Rosenfeld at the University of San Diego, was pioneering a lot of studies on transcriptional regulation. Although his lab was not really working in neuroscience, but I was lucky enough to be offered a postdoctoral position to do exactly what I wanted, learning a lot of advanced methods to study gene transcription. And so I moved across the pond and I moved to San Diego to do my postdoc. And then after that, I actually opened my lab at UCSD, and I'm now an associate professor at the Department of Psychiatry, where I keep working on gene regulation, and mostly in the context of the neurobiology of addiction.
Yeah, and we will talk about the exact science that you're doing there in a minute, but let's hear it from Jessica.
Yeah. Hi. So I guess I've always been exposed to biology because that's the field that both of my parents work in. But actually, when I was younger, I didn't really want to do science. And when I went to college, my parents also encouraged me to do something besides science, something different from them. And so I started college as a business major, actually. And I did that for about a year, but then I kind of felt like my brain wasn't working hard enough. I guess I missed doing working with numbers and doing science and things like that. So then I started looking into changing my major after my first year in my bachelor's. And so I ended up switching my degree to biomedical engineering at the end of my second year of college. And, I continue to take courses there, but I think I realized that I specifically liked working with numbers and doing the math. And so I decided to look into computational biology. And so I was able to join a lab that did some cancer research. It was a new lab at my college. The professor had just started in January, and I contacted him in February. And so I started doing some very simple computational analyses of cancer genomics data. Then I also had the opportunity to do a summer undergraduate research program in Boston. That was the summer before my senior year of college. And there they really kind of like encouraged you to, think about applying for PhD programs. They taught you a lot about what it takes to apply to PhD programs. And I think getting my first experience just doing full-time research, like not, also having to do classes too, but just, every day I wake up, I go to the lab, I work all day, and then I come home, and I was, living in a dorm with other students in the program who were also doing full-time research. I think that was a really cool experience, and I really liked being in that environment. And so when I got back to college, I decided to apply for PhD programs in computational biology and bioinformatics. And that's how I ended up at UCSD doing my PhD. And I actually joined a new lab again. I joined Dr. Graham McVicker's lab. I guess I enjoyed working in that small environment with like a new professor and young professors. And during my second year, I applied to a Fulbright in Berlin, and I got it. So during my third year, I was kind of working on my research project over in Berlin as a visiting scientist. And the project there focused on analyzing data from a single-cell CRISPR regulatory screen. And so through that, I got some experience working with single-cell data. And so when this grant that my professor, Dr. McVicker and Francesca applied for came through, and they wanted to put someone on the project, Graham thought of me, because I had experience working with single-cell data, and the whole point of this project was to generate these novel single-cell data sets and to analyze them. And so that's how I got put into contact with Francesca, and also Dr. Abraham Palmer, who was also one of the co-authors on this project. And that was actually right before the pandemic started. So when the pandemic started, I had to suddenly come back to America, and then this project kind of became my main focus from there on out for most of the rest of my PhD.
Yeah, and then what work you were doing together we are coming to during this episode. But let's come to the science part of this episode that centers around the molecular underpinnings of human addiction. Francesca, let's start in 2015. There, I think your work on epigenetics in the brain started. The epigenetic mechanisms linking changes in neural transcriptional programs to behavioral plasticity remain largely unknown. In your first paper that was published then in this paper I was referring to that was published in 2015, you identified the epigenetic signature of the neuronal enhancers required for transcriptional regulation of synaptic plasticity. Can you talk about this study and also how you ended up in California? You just shed a little bit light onto this after your PhD in Italy.
Yes, absolutely. So as I mentioned before, right when I was ready to start a postdoctoral project, I was looking for learning and, on my skills in more advanced methods to study transcription. And particularly at that moment, I was interested in learning and memory. And so more of the mechanism that regulate just our normal behavior. And, the brain is incredibly complex, and gene expression play a very key role both in the development and functioning. And I wanted to continue studying gene expression regulation to gain insight in how not only the brain develops, but mostly in how the brain responds to new experiences. And that's what happens when we learn something new, we experience something new, and we retain that information. How that information is encoded is what I wanted to understand. And I knew it was encoded in our genome and in the interplay between the genome and the environment. And that's where the epigenetics mechanisms play a big role. And that's where I moved to California to work in the lab of Michael Jack Rosenfeld, who was using these new methods like chip sequencing to study how different proteins and different histones, different proteins bind to specific region of the genome and control the expression of different genes, and how histone modification also affect the way genes are turned off or turned on. And as I mentioned before, the lab wasn't working really in this field, but that was the fun part. I was given this opportunity to do what I wanted and to be free to explore this new avenue of research. So at first, I had to set up the entire, operation of getting of studying the brain, and I was doing that by both looking at neurons, which are, the building block of our brain, and how they respond to stimulus that would sort of reflect the way our brain responds to new environment. Every time we're exposed to something new, there are some specific brain region and neurons in specific brain regions, such as the hippocampus, that will fire and respond to these new experiences. And I was modeling that in a dish. And so I identified a specific molecule, renin, which regulates how these neurons respond to a new experience. And so I was using these methods to understand what genes are going on and off. And this study showed us that there are specific regions of the genome which are called enhancers and are particularly sequence that control which gene turn on and off in specific cell types. And that was fascinating for me that we were getting this very detailed understanding of not only the signaling molecules, but also the specific DNA regions of the genome that before were considered junk DNA or regions that non-code for genomes, for genes, for proteins, and so they don't have a very clear function. But we were learning that they actually are central to regulate the way we learn and the way we retain information for a long time. So that project really looked at how we encode new memories and how we retain those memories for a long time. And that was regulated by renin.
And what kind of marks does this influence? So are these the classical enhancer marks, or were there different ones?
At that time, I looked at, in that specific project, we looked at just known enhancer marks, such as H3K27 acetyl, for example, and other known markers of activity, of active transcription. But actually, in a parallel project that I worked on, always focused on mechanism of learning and memory, we studied another molecule, histone demethylase, LSD1, which we found that was a very fun project where we found that a splicing isoform of this enzyme, which changes only 8 amino acids of this protein, which we call neuronal LSD1, because this isoform was only expressed in neurons and at the inclusion of an exon that encoded for 8 amino acids. But those 8 amino acids, they landed right in the enzymatic pocket of this site of this enzyme. And what we find was very fascinating, this change in the enzymatic site of LSD1 changed completely the substrate specificity of this enzyme. And so in that case, we looked at new marks that were associated with learning and memory, specifically LSD1 demethylase removed methyl groups from histone 3 or histone 9. But now we discovered that a new substrate, which was histone 4 lysine 20, was a new substrate of this enzyme and was involved in the way the neurons again responds to firing and the way the brain responds to new experience. And we discovered that this histone H4K20 monomethyl decorated the gene body and was controlling a specific step of transcription, which was elongation. So that was another project where I worked on epigenetic mechanisms.
Now, this seems to be a very unique feature of this protein, right? I mean, I've heard that there are splice variants of proteins, but that this specific variant of splicing changes like the enzymatic activity and to a complete different histone modification seems to be very unique and very interesting, right?
Yes, it was very interesting, but we were very relieved that other researchers at the same time, they were studying this neuronal isoform, and they also found that this switch in the enzymatic activity was really important in neurogenesis, in neuronal development. So although very unique, it is not uncommon that splice variants regulate the function of many important neuronal proteins. So I think there's a lot more to discover in this space, and looking at how splice variants change the function of proteins.
Yeah, you then started your own lab and started to look into drug addiction in a broad sense. First, you looked at long-term effects of cannabis use. However, the factors that contribute to the long-term detrimental effects of cannabis use are not so well understood. You then studied how relin, the protein you just mentioned, deficiency, influences the behavior of mice exposed to cannabis during their adult life. So what did you find there?
Yeah, when I started my own lab, of course, it was not a coincidence that I was working on what you just described. And so coming from, starting from my work on learning and memory and reeling, I wanted to understand how not only this signaling pathway regulates normal behavior, but how potentially disruption of this pathway can influence pathology. And so I was looking at a model, system or a disease model that would be good to explore this hypothesis that disruption of reelin signaling and consequence disruption of memory can contribute to disorders. And the mouse model of chronic exposure of cannabis in adolescent brain was the right answer for me, because cannabis, as many other psychoactive drugs, are very strong modifiers of behavior, and that's what drew me towards studying neurobiology of addiction, besides being such an important public health concern, an issue right now in the United States and worldwide. But I was fascinated by how psychoactive drugs are such strong modifiers of behavior. And cannabis specifically, and its effect on the adolescent brain, can modify learning and memory, which was my major behavioral domain, let's put it that way. And when we looked at how, reelin signaling, so more of an internal signaling, interact with an external environmental factor such as cannabis exposure, We learned first that there is an interaction between these two signaling, which probably means that reelin interacts with our endocannabinoid signaling, because we have an entire, signaling pathway and an entire system of molecules, including receptors and enzymes, that are able to produce and respond to cannabinoid-like molecules, which are our endocannabinoids. And endocannabinoid signaling is absolutely important during development, such as reelin. So that's where my hypothesis came about, of how these two signaling pathways that are important for learning and memory and development could interact. And I used cannabis, which is very widespread use by teens. And so there's a lot of concern on how not only abuse of these drugs and the new high potency strains of cannabis and synthetic cannabinoids, how they affect adolescents. And so in our first work, we looked mostly at the behavior, because although there were a lot of studies, none of those previous studies have looked at how cannabis changes behavior during adolescence, where we manipulated the reelin signaling pathway. And we found that first, as others had shown, chronic exposure to cannabis can reduce memory performance, and that in the animals, mice that don't express reelin or express lower level of this protein, some of the behavioral features were further compromised. And then we looked more closely also at what are the gene expression changes that are induced by chronic use of cannabis during adolescence, and they might underlie these long-term detrimental effects on brain functions. And we did another project using a transcriptomic profiling in different brain regions of the brain. And we found that many gene co-expression networks were correlated with THC, which is this psychoactive component of cannabis, and were also correlated with the reelin genotype, suggesting again that these two pathways overlaps. And particularly, we found that VTA, the specific region of the brain where there are the dopaminergic neurons that respond to most psychoactive drug. VTA was particularly affected, but also other brain region of the brain reward system, suggesting again that this psychoactive drug, they can manipulate the way we perceive reward, and they could lead not only to the memory deficits and social interaction deficits that we had observed before with behavioral studies, but they also suggested that this chronic adolescent exposure to THC can affect the brain reward system. And so right now in the lab, we're kind of looking more at that side of the story. What are the pathways that are affected by cannabis during adolescence, and they can lead to cannabis use disorders?
You also checked those gene expression networks in different sexes, right? In males and females. What are the differences, and why are there differences?
Yeah, that's a very good point. We are very interested in understanding sex differences. And most of the previous literature in animal research, also not just in the field of addiction, has mostly worked on males. And there's a lot of misconceptions because of this type of experimental design. There's a strong need to do more research using females because the biology of sex differences can affect behavior and can affect, therefore, the way males and females respond to treatments in different pathological conditions and therapeutic settings. And so there's a lot of drugs that are designed based on studies that have only been done in males. And so that was one of the motivations for us to look at sex differences in the way male and female mice respond to chronic adolescent exposure to THC, because already epidemiological studies showed that there are sex differences in the way teenagers respond to cannabis, and there were already some evidence that females might be more sensitive. And we found some of those same evidence in our animal studies, where we found that some specific behaviors were more heightened in females, such as risk-taking behaviors, and some other features were more pronounced in males. We still don't understand what are the molecular mechanisms driving those sex differences, and those are probably linked again to how the endocannabinoid signaling behaves differently and in the two sexes, and mostly because endocannabinoid system is so important during sexual behavior and also development of reproductive system. But we need to do more work to understand the mechanism behind those sex differences.
So next to THC and cannabis, you also investigated cocaine addiction. So why did you also focus on cocaine addiction, and what is the difference between cocaine, THC, and both addictions?
Yeah. So, most psychoactive drug and addictive drugs share some, basic mechanism of action. For example, cocaine or THC or opioids, they all share one common mechanism, which is increasing the level of dopamine in the brain after a person is exposed and consumed the drug. And that, basic mechanism induces a series of downstream effects in the brain. But at the same time, those drugs work differently because they bind to different receptors that are expressed in different parts of the brains or the body. And so it's fascinating how trying to understand those commonalities and those differences. The reason why we focus on cannabis is one of the main reasons is because it's widespread use during specific critical periods of development, such as adolescence. And so we use that system to study mostly the effect of cannabis and drug of abuse on the adolescent brain. But cocaine is another, a very widely used drug, and recently has been implicated in a concerning rise in overdose deaths. And that's one motivation why we focus on cocaine addiction, because it's a very serious health issue right now and societal issue. But again, we are finding some common mechanism between different type of addictions, but at the same time, we're interested in understanding what makes each drug affect the brain differently.
So let's come to the work that you both have been involved together, and let's involve Jessica again, since you have been quiet for a while. There you did single-cell work where you generated an atlas of single-nucleus gene expression and chromatin accessibility, so right in the ballpark of our epigenetics interest. Could you maybe talk about this story and what this atlas helped you to find?
Sure. Yeah. I think Jessica mentioned before that I was collaborating with Graham McVicker, a graduate advisor, and for some time, Graham and I were discussing about how to fill some critical knowledge gap in this field. And there were several, barriers that we found in the current literature and in the field. One was that most studies in the addiction field, especially looking at gene expression changes in the brain, were using inbred strains of rats or mice. And so that was a barrier that we wanted to overcome, because using one specific genetic strain does not reflect what happens in the human population. And so there was one reason we partnered up with A. Palmer and Olivier George, who are using this outbred population of rats that are genetically diverse and are closer to the human population. Another barrier that we found was that most of the gene expression studies were focused, or epigenetic studies were focused on bulk profiling. So we didn't have the cell type specificity that is so critical, because as we know, the brain is extremely complex and heterogeneous, and we wanted to, delve into this cell type specificity in terms of how different cell types, neurons or glial cells, respond to exposure to drugs or contribute to susceptibility to drug addiction. And lastly, most studies were using animal models where the experimenter would administer the drug by injection, so it was a forced exposure. And again, that did not really reflect what happens in the human population when people decide to take the drug. And so to better model that motivational aspect of behavioral traits associated with addiction, we partnered with Olivier George at UCSD, where they developed these more comprehensive protocols of self-administration of drugs in rats. So in this case, the rats were trained to self-administer drugs. So they would decide when to take the drug, how much drug they wanted. And we decided to look at what happens in the brain after a long abstinence, not just after acute exposure to drug. And that's because one of the features of addiction is the high rate of relapse. Even after a year of abstinence, some individual, not all, but some individual can relapse. And that's one of the critical problems to face when we need to try to understand how can we treat these devastating disorders.
So you were you were basically looking for permanent changes, right? So that really last in the brain.
Yes, changes that persist. And so we designed this project based on these limitations that we wanted to overcome. And of course, it was a challenging project, and we needed someone who was able to take up this challenge and lead us in the right direction. That's when we recruited Jessica, and she can tell you more on her contribution to this story.
Yeah. So, for me, it was very exciting to get a bunch of novel data that's like designed to answer the question at hand. Because actually up until this point in my PhD, our lab was small and it was primarily a computational lab. We did not generate a lot of our own data. So a lot of times you're kind of like scavenging for data, using like data from previously published studies and trying to repurpose it or reanalyze it. And sometimes you find that your data that you're working with is not sufficiently powered to answer the question question that you're trying to study, or, you're like, oh, if only I wish it were like this. So it was really exciting to work with a group who was designing data specifically for the research question. And I also had a lot of it, like single cell data is still relatively new. I mean, it's very popular, but it's quite expensive to generate. And there's technical challenges associated with both doing the experiments and analyzing the data that you don't get with traditional bulk sequencing. And so, it was kind of fun to get this data and then learn about this question. I'd also never really worked with neuroscience or learned about how animal models work and all the important nuances that come into play when you're working with like these behavioral animal models. I think Francesca talked a lot about how there are certain aspects of this project that were unique to our project that kind of like really hone and refine the question that we're asking. For example, because we let the rats self-administer, that has different implications for the how we interpret the findings, and also because we studied long-term changes. We're not just looking for changes associated with addiction. We're looking for changes associated with addiction that can predispose an individual to relapse, which is one of the main obstacles in treating addiction. And now, when it came to analyzing this data, as I mentioned with single cell, there are a lot of challenges because the data doesn't look quite the same as bulk sequencing data. Because with bulk sequencing, you're getting information about the expression of a gene from an entire tissue sample. So you simply have more material to work with and to detect gene expression or these biological signals from. When you work with single cell, you're individually profiling individual cells. And so a cell is very small, so there's simply going to be less biological material to analyze. So the data can be very noisy. So, that required us to think carefully about, how to analyze the data, and also at every step we had to be very rigorous with our quality control and think about what all the numbers mean and how we should take this into account when moving forward with the next step of the analysis.
Sorry to interrupt, do you get like a black and white picture then in single cell expression data, so either the gene is off or on, or do you also get like intermediate, so the gene is like 60% on?
You do still get like individual reads like you do from a regular RNA-seq data experiment. You just have less. So there's going to be more like zero counts. There's going to be a lot of genes where the expression might not be detected because there simply weren't a lot of reads to capture during the RNA sequencing experiments. So that's done. So it's not like going to be a binary vector. With ATAC sequencing, I do think at least sometimes people will binarize the data just because there the data tends to be even sparser when you're looking at single cell ATAC-seq compared to single cell RNA-seq. But, we still treat it as count data. We still look at the discrete counts and then measure that. So we weren't working with off or on metrics, which, gives us, I guess, more detail to work with when trying to interpret the data. But yeah, because we had single cell data, we were then able to characterize the cells. That does require a decent amount of manual labor. I work closely with Francesca for that. She's the expert on the neuroscience part of the project. So, after doing careful profiling of the expression of different genes in all of the cells, and grouping the cells based on the similarity of their gene expression profiles, we were then able to basically use pre-existing knowledge of what cell types we expect to find in the amygdala and, their known gene expression profiles, like what we already know about how these cell types tend to express. And through that, we were able to manually annotate this atlas of single cell RNA sequencing data that we had. And interestingly, we also did single cell ATAC sequencing in the rat amygdala, but they weren't from the same rats for the most part. And this was
because not a multiomics, a true multiomics experiment, because you didn't do the experiments from the same mice and the same cells, right?
Yeah, exactly. So, we couldn't simply just use the same cell type annotations. So we used a label transfer approach to basically map the cell type labels from the RNA-seq to the ATAC sequencing data that we had. That made it possible for us to therefore characterize the cell type populations present in our single cell ATAC sequencing data as well. And then in all the downstream analysis, we were therefore able to perform all of our analyses at the cell type specific level. So we could say, like, when we're looking for differential gene expression between, cells that came from the rats that showed a high addiction index compared to the cells that came from the nuclei, I should say, because we did single nucleus sequencing in the rats. We were able to identify differences that were specific to individual cell types. This could be interesting because some genes might only show differential expression between the high and low addiction index rats in like neuronal cells, but not glial cells. And in doing something like that, you can get a better understanding of the molecular basis of the disease. You don't have to say it's just we identified this in the amygdala. We can say this is specifically coming from this particular cell type in the amygdala. That can also help you not just gain a better understanding of the disease, but also develop more targeted treatments for it.
And what kind of differences were you able to find between like the acute cocaine uptake and then like the long range effects? So how much genes were changed or how much cell types even were changed?
So one of the main findings from our paper is that we saw that among the genes that were significantly differentially expressed between high and low rats, the ones in the neuronal cell types mostly tended to be enriched for genes belonging to pathways related to cellular, like metabolism. And based on this, we did several follow-up experiments, which maybe Francesca can talk about, and how the translational impact of that.
Yeah, that was one of our major findings, that oxidative phosphorylation pathways, which are related to how the cells produce and use energy, they were affected across cell type, mostly in neurons, but across different subtype of cells. So that meant it was a strong signal. And that also suggested us that an altered metabolic state of the cells can be perhaps targeted by, for example, a pharmacological intervention to try and re-establish the normal functioning of these neuronal cell types. And so what we did in some of the following follow-up experiment, we used a small molecule that inhibits a metabolic enzyme, which is called glyoxylase 1. And this enzyme is involved in the clearance of methylglyoxal. Methylglyoxal is another metabolite that typically the cells try to get rid of because if it accumulates can cause senescence. But in the brain, some recent discoveries have suggested that this metabolite can actually affect neuronal transmission, particularly GABAergic transmission, because methylglyoxal can act as a agonist for the GABA-A receptor. And so this knowledge suggested us that perhaps if we affect the way the cell metabolize glucose and therefore glycolysis, we might affect directly GABAergic transmission, which is so important, especially in the amygdala, for the normal functioning of this brain region, but also has been implicated in addiction. So we use this molecule to manipulate the activity of the enzyme, which is responsible for the metabolism of this GABA agonist. And indeed, we showed not only the GABAergic transmission was altered between the rats that have high or low addiction index, but we could reverse this enhanced GABAergic transmission by treating slices of the amygdala with these small molecules. And we also use this small molecule in vivo in the rats by injecting rats that were either more predisposed or less to become addicted with the GLO1 inhibitor, and we could also revert the higher relapse that we can observe in rats that have taken a lot of drug and that have these high addiction indexes. So in a way, our transcriptomic analysis give us a cellular snapshot of how the brain of rats that have high addiction index are compared to the low addiction index. And they showed us that altered metabolic pathways related to energy consumption are a strong hallmark of the cellular phenotype. But the cellular phenotype then affects not only neuronal transmission, in this case GABAergic transmission, but they also affect consequently behavior. So we were able to link differences in gene expression all the way to behavior. But a question for us remained, which was, is this an effect of cocaine exposure, or is this an effect due to pre-existing genetic differences? And that's where Jessica did more analysis that maybe she can share to try to understand, what are the underlying mechanisms that contribute either to predisposition to develop addiction or progression of addiction due to drug consumption.
Yeah. So, normally if someone wants to study genetic predisposition or genetic risk for something, you would do like a genome-wide association study where you would basically sequence a population of individuals that, you kind of divide them into a case and control, and then basically look for associations between certain alleles and the trait that you're trying to study. So, if we wanted to just study like a rat's genetic predisposition for addiction, we would have had to put a lot of rats through this protocol, sequence all of those, and then do an association study. And, we simply didn't have that scale, that number of rats available. So that approach was out. We couldn't do any sort of genetic association study to see, do the rats in our study have, certain variants that put them at risk for being high addiction index or low addiction index. Our collaborator, Abe, he is working on doing kind of like these rat GWASs, but they just weren't ready yet at the time that, we were publishing this paper. And so we had to come up with a workaround that, would answer this question with the data that we had available. So what we did do was we tried to because we do have the genetic profiles of all of the rats in our study. They are genetically diverse and they were all fully sequenced. And so what we did is we had the observed gene expression, and we were curious how that would deviate from their expected gene expression based on their genetics. So our collaborator, Abe, they do have enough data to develop some predictive models of a rat's gene expression in certain tissues based on their genetic profiles, basically based on the alleles that they have present. So we leveraged some of those models working with Daniel Monroe from Abe's lab. He's a postdoc. And we basically imputed or predicted gene expression for all of the rats in our study based on their genetic profiles. And we compared these predicted gene expression values to the observed gene expression values. And what we found was that it was reasonably consistent. And this scaled as we increase certain stringency metrics that basically measure how well the predictive models perform. And so what this tells us is that genetic influence does play a role in affecting the changes that we observe from our analysis, if that makes sense.
Definitely. Francesca, you're looking like you want to add something.
Oh, no, yeah, I was saying that was a very, in a way, we expected that genetics plays a role, but that was probably one of the first time that there was evidence at a single cell level that we can more confidently say that those gene expression differences are due to genetics. We were, able to provide this evidence, which was important for us to say the sum of these long-term changes that we observed after cocaine self-administration, they might be due to genetic predisposition, to pre-existing differences. And that was important because, in a way, there's a lot of stigma around, addiction. And it's important to remember that addiction is or like other complex psychiatric disorders, where it's not just the choice of the person to become addicted, but it depends also on some genetic factors. And so that, in a way, is important to remember that genetic factors can predispose some individual to be more motivated to consume drug. And then even if they quit, they might be more predisposed to relapse in specific situations.
So I think this study gave you a lot of material to do future work. So imagine you both are due to submitting a grant proposal tomorrow. What would you write into that?
Oh. Huh.
At least grants submitted yesterday and a month ago. We've been thinking about this now for a while. And definitely we want to study more rats. As Jessica said, some of the limitation of our study in this context, when we are trying to understand or disentangle the contribution of drug exposure versus genetics, would benefit by having a larger cohort of animals. And so that's, and to use some, predictive modeling and like polygenic risk score to understand better the individual vulnerability to addiction. So definitely this study inspired us to think more deeply on how can we study the genetics of addiction and how those genetic factors affect gene expression and chromatin accessibility.
So that's more moving away from the pure epigenetics, but more to the primary segments of the DNA?
That's one direction, but definitely we haven't talked about too much about the results or implication of the chromatin accessibility studies in our work. But from that angle, we also learned a lot. There is a very profound difference in the, let's call it epigenomic profiling or chromatin accessibility profiling, that is very cell type specific. For example, we found that excitatory and inhibitory neurons of rats with a high addiction index versus low addiction index, they look completely different, if not opposite. In a way, the excitatory neurons showed way more site accessible, while the inhibitory neurons showed that a lot of sites were less accessible. And with the help of also another student from Ground's lab and Jessica's work, we found that specific transcription factors are enriched in those affected sites in a cell type specific manner. And that's one other next step, understanding how those transcription factors' activity might affect the chromatin landscape of different cell types. And that's very challenging because while we can study gene expression and chromatin accessibility at the single cell level, studying transcription factor binding at the single cell level is still, a very new approach that is not yet very accessible, for example, like with the multi-omic kits that you mentioned. And so that's something we're looking into it as well.
Yeah, I was just going to add on, for that study, what we were really looking for was actually enrichment of transcription factor motifs in the regions of the chromatin that were differentially accessible between high addiction index rats and low addiction index rats. And we did this in a cell type specific manner. Now, the interesting thing about transcription factor motifs is that a lot of related transcription factors will recognize similar motifs. And there were so many of them in the database that the student that we were working with, Aaron Ho, he was a software engineer in Dr. Graham McVicker's lab, who's actually now applying for PhD programs. He helped a lot with this paper. He grouped transcription factors that had similar binding motifs into families, and then we were looking for enrichment of those common binding motifs. And so what we came up with a hypothesis of binding motifs that are enriched in those differentially accessible regions. But we would need further experiments to confirm whether those transcription factors actually are binding in those differentially accessible regions or not. And so that's what Francesca was saying is difficult, right? Like we came up with these cell type specific hypotheses of transcription factor binding, but there isn't really the technology yet for like single cell chip sequencing, which was someone would typically do to study whether or not a protein is bound at a certain region in the DNA or not.
Yeah, maybe the new methods, cut and take it, cut and run, can help investigating this then in the future. But Jessica, since you switched coast, right, you moved from the West Coast to the East Coast and are now in a different lab. Are you working on the same things, or what are you going to investigate moving forward?
Yeah, so I am actually working in a somewhat of a different area now. I'm doing a postdoctoral research position at Cold Spring Harbor Laboratory in Dr. Peter Kuh's lab. His lab focuses on developing machine learning methods for analyzing DNA sequence data and trying to understand its regulatory function. I did this because I wanted to get more experience building and developing machine learning models and specifically deep learning models. And so that's what I'm working on right now, trying to work on developing interpretable machine learning models for genomic sequence data so that we not only are we able to make predictions about the effects of a DNA sequence on biology, but also to get a better understanding of what these intricate deep learning models are learning, because that'll give us a better understanding of how to actually interpret the regulatory genome. Something like that could also be interesting, though, if we wanted to try to study some of these questions. Like if you could train a machine learning model to actually predict transcription factor binding, not just the presence of a motif, but whether or not a transcription factor is binding in a region, that could be useful for, filling this niche that we currently don't have the technology to answer. Okay.
So for the last 57 minutes, we have been on a journey through your both scientific careers. Did we miss something important, or would you like to add something?
Definitely, I want to add that although all these projects, presented a lot of challenges, it was a lot of fun to work together with a very bright student, Jessica, but also a bigger group of collaborators. This was really a project where teamwork is the only solution to address big challenges. Because, for example, we put together not only different expertise and different experimental models, from the outbred rat genetics to the self-administration studies, to the computational modeling and to the profiling and sequencing single-cell genomics methods, that doing it in one lab is definitely possible, but it might take too long for a student to graduate on time, or, so it was definitely the bright side of this was having the opportunity to collaborate and interact with scientists from different disciplines. And so I wanted to add that.
Okay, so then thank you, Francesca and Jessica, for your time and for being on the show.
Thank you. Thanks for having us. It was really fun to talk about our paper with you. Hopefully people can learn a lot from this podcast, and it makes everything more accessible to people.
Thank you. It was really fun as well to spend our morning with you.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.